needs by making small changes to something it knows. Now, we thought that maybe we should have something like that with the endocannabinoid system as well. And so we looked at compounds which may be closely related to anandamide and 2-AG. Both anandamide and 2-AG are derivatives of a particular fatty acid called arachidonic acid. So we looked at additional arachidonic acid derivatives, and yes, indeed, we found in the brain arachidonylserine, which you can see on the slide, which is closely related to anandamide, 2-AG. It does not cause any of the effects that we know, the psychoeffects of THC or anandamide, but is most definitely acts on the vasoconstriction. So here we see again the same type of effect that we have seen with other biological systems. Now we went ahead and took this particular compound and tried to see whether it also helps lower the damage done by brain trauma, as 2-AG does. And indeed, this is what we see. We see that 2-AG and RAS, arachidonylserine, act in the same way. You can see the blue line and the green line, which are, one of them is the effect of 2-AG, the other one is the effect of RAS, and they are completely different from the effect if we don't give any compound of the vehicle. So the body, or brain at least, tries to find many ways to lower brain trauma by using endogenous cannabinoids and closely related compounds of the same type. Now, I will not go to additional effects of 2-AG, or I'll go to some of the effects of THC. Well, we know pretty well that 2-AG acts on appetite and feeding. We show that if we inject pup, an animal that was just born, we inject it with an antagonist to 2-AG, and these are of course known, then the pup, although it's next to the mother, does not suck. It does not know that it has to suck. It sucks because it has an endocannabinoid system, anandamide, 2-AG, and these, in some way, we don't know exactly the way, causes the animal to go and suck milk. If we block the system, the animal does not suck, and it will die within a few days. But if after a day or two, we give a large amount of anandamide or a large amount of THC, so we can kick out the antagonist, then the pup goes back to the mother, starts sucking, and survives. So here we have a definite proof of how anandamide, 2-AG, and maybe other compounds act on feeding and suction. Now, I mentioned the antagonist, and the antagonist, as it blocks feeding, blocks the appetite, a company thought that, oh, well, it will be a good compound to act against obesity. People will eat less. Well, and the company spent a huge amount of money and put the compound on the market, and it had to withdraw it because they didn't take into account that anandamide, and this is an antagonist to anandamide, the endocannabinoid system, anandamide and 2-AG, also do other things like they lower anxiety. And if we block the system, then we help the obesity on one hand, but on the other hand, enhance anxiety. And people that do have anxiety may have a very severe anxiety. And I understand that there were a few cases of people that wanted to commit suicide, so the company took out the compound from the market. So we have to know, to take into account the many effects that the endocannabinoid does. Now, I will go to cannabidiol, which, again, as you know, is a major compound found in cannabis. And cannabidiol, and here the two structures that I showed you before, the THC, which found in 1964. As a matter of fact, the structure of cannabidiol we elucidated even before that 1963, which is 51 years ago. Now, cannabidiol is a very, very interesting compound. And we started by looking at the effects of inflammation on cannabidiol. And we found both in vitro and in vivo that it lowers the amount of TNF compound that causes inflammation. It lowers the amount of NO that the body forms and which is again involved with inflammation. And it also lowers the amount of reactive oxygen intermediates. So we thought that maybe it should be active in animal models of inflammation. And with a group in London, we went ahead and indeed in rheumatoid arthritis, we gave cannabidiol to these animals. And we found that at a certain level, 5 milligrams and then went up to 10 milligrams at 20 milligrams. And we found that there is at a very low dose, of course, cannabidiol does not work. It works at a certain dose. But if we go to 20 milligrams, then the effect goes down. This is a biphasic effect, which we have seen with, in many other cases. So in inflammation as well, we have a biphasic effect, very important. After going to reaching a certain level, giving more material can lower the effect, or the effect may disappear completely. We looked at whether the cannabidiol acts on synovial cells. You're aware that the synovial cells in mice, as well as in humans, produce large amounts of TNF. And as I mentioned before in a second, that TNF concentrations of TNF go down. Well, indeed, we found that in mice, in the synovial cells, which are very important for the production of TNF, these cells produce much less TNF. So this can probably prove why cannabidiol acts. Then we went into something else entirely, and this is diabetes type 2. Now, we went into diabetes because rheumatoid arthritis is an autoimmune disease, and diabetes type 1 is an autoimmune disease. So we thought, well, maybe we should look at autoimmune diseases. And this was then one of those. There are mice that sporadically get diabetes type 1, and 100% of them are sick after 12, 14 weeks, start getting diabetes, and they die usually, of course, much sooner than the healthy animals. So we try to find out whether cannabidiol, which acts on one autoimmune disease, whether cannabidiol can act on another autoimmune disease, diabetes type 1. So we gave the material not from scratch, but we gave the material, the cannabidiol, only when the animal had already shown some of the starting effects of diabetes type 1. And this is after 12, 14 weeks. And you can see on the slide that while animals that are not given CBD, almost all of them, 80% of them at least, or sometimes 90% of them, develop the diabetes, while the animals that got CBD, and this is here the line up there, shows that only 30% got. So here we have something that seemed to act extremely well, diabetes type 1, and I assume that cannabidiol can be used in diabetes type 1, which is a difficult disease to treat. So we gave these animals to our histologists to look for the effects of cannabidiol, And they looked at the number of islets that produce insulin. They looked at the islets that were ruined entirely, and they looked at many other things. But to just look at the last part of the slide, which says percent of intact islets, those cells that are very much involved in diabetes. And we found that in the control, essentially all the islets were, there were no intact cells at that point. Those that got the vehicle, again, only 13% were intact. But those animals that got CBD, 77% of the animals had intact islets. So here we have something very basic, which we see. I'm not sure that we know the mechanism of that, and I'll try to speculate on the possible mechanisms. We have seen that cannabidiol, our group and many other groups, acts on anxiety, on epilepsy, on schizophrenia, and we actually used cannabidiol against epilepsy in adults. We published that about 35 years ago, that cannabidiol at high doses, 200 to 300 milligrams a day, lowers the number of epileptic attacks. Unfortunately, this paper was disregarded, and up till this very day, there are no major trials with cannabidiol in epilepsy, although we know that cannabidiol acts on certainly in children in epilepsy, and there are now quite a few groups that are trying to do clinical trials. Why was our paper of 35 years ago disregarded? I can't give you an answer. I really don't know. Now, there is another paper that was published quite recently with cannabidiol in schizophrenia, and it was found that cannabidiol parallels a known drug in schizophrenia as good as the known drug, with one exception. The known drug causes side effects. Cannabidiol does not. So are we going ahead with cannabidiol in schizophrenia? We had previously shown that in a single patient, and the paper I was telling you about was done on quite a lot of patients. So we have a pretty good basic knowledge that cannabidiol acts on schizophrenia. The doses needed are very high, about 800 milligrams. But maybe one can have cannabidiol derivatives, which are more potent, and then we can lower the dose needed. Now, there are a lot of effects of cannabidiol, and I just wonder whether cannabidiol acts through some basic regulatory mechanism. One of these basic mechanisms is epigenetics. Now, epigenetics is the study of the changes that take place on the DNA, but the DNA itself, the basis of the DNAs are not changed. Only there are changes on, if you wish, on the surface of the DNA, and by making changes on the surface of the DNA, one can block or activate a particular DNA. And for example, the body knows how to methylate, put a methyl group on parts of the DNA. I would not go, of course, into details here. And this methylation blocks the effect of that particular DNA. And indeed, there is at least one example published by a group in Italy by Professor Maccarone in Rome. He found that in a certain skin disease, there is some methylation by cannabidiol. Cannabidiol acts on this disease, lowers the effects of this disease, and it causes methylation on the DNA